So
That's become a growing concern since that salmonella outbreak linked to tomatoes. It has now made nearly 900 people sick in 36 states and the districts since mid-April. It's the largest salmonella outbreak linked to fresh produce in U.S. history, and the government still can't pinpoint the source. With each passing day, Kelly Cobiella reports the human and economic cost is growing.
You want a bowl or a cup?
In the heart of Florida's tomato country, restaurant owners are worried.
It's been paying bills every week instead of,
Hoping that
Yeah.
the money's there?
yeah.
Locals are frustrated,
Our economy's been affected by that, the growers have been affected by that.
and growers are fighting off their anger.
I've got a sour taste in my mouth about it.
Because nearly 3 months after the first case of Salmonella St. Paul, people are still getting sick. At the Tomato Cafe in Havana, Florida, owner Cheryl Moore has been trying to reassure customers about her key ingredient for weeks.
Sure, because you're wondering if you're gonna be the next place that it happens at, because you don't know what's caused it.
It's frustrating. It's frustrating for FDA.
The latest case was reported June 20th, 12 days ago. And because a tomato's shelf life is about 2 weeks, it means the source of the outbreak is still out there. In fact, the FDA isn't even sure it is tomatoes. They're now looking at other produce often served with tomatoes, though they won't say what. Today, the FDA tried to put the scare into perspective.
850 cases is a lot. But 850 cases in a population of 300 million. We need to be thinking about, well, what are we talking about here of a crisis?
Critics don't buy that.
It really is criminal that FDA has not acted sooner to get regulations in place and mandatory tracing systems for these products.
Grower Virgil Butler is still struggling to sell his crop. Sales of tomatoes in June were down 40% nationwide.
Do you think the FDA failed in its mission?
Yes, I really do. And I think they don't know how to fix it. The damage has been done. How they want to fix it now?
Butler isn't waiting around to find out. After 30 years, he's getting out of the tomato business altogether. He's decided to grow grass instead. Kelly Cobiella, CBS News, Quincy, Florida.
okay, this gives an idea, okay, about outbreaks, and it's not just people that are getting sick, it's the economy, okay? It's really the main problem, because with people, if you have some outbreak in tomato, then for the public and the entire vegetables are contaminated and they may have a problem. As for example, the grower was saying before, okay, I didn't exactly, it's like big concern about this. And if we see the outbreak in 2014, so this data came from CDC, United States, but in Europe also we have outbreak in vegetables. 2012 we have mangoes, cantaloupe, and we have of course poultry and beef. 2013 cucumbers, okay, and last year nut butter and sprouts and cucumbers again. So vegetables are involved in this process. Okay, so our goal was to try to figure out which are the genetics factors involved in this proliferation and to study the tomato salmonella interaction from a molecular point of view. Okay, so salmonella can proliferate in tomato. It's proved can proliferate from the root and can go up, okay, through the stem. Salmonella can stay in the flower, okay, and there are evidence in which scientists had inoculated the flower and they found that salmonella in the tomato. So there is this transfer, okay. We don't know at which level, we don't know if you inoculated the root with a lot of salmonella, you will find all the tomato full of salmonella, for example. We don't have this proof yet, but if you have an outbreak involving half the United States, that probably your entire product is full of salmonella. Okay. So this is a picture from salmonella stem. Okay. And so basically this is the cross section of this part. And this is a salmonella in XLD. This is XLD plate. It's a patch plate in which we can grow salmonella. I took this picture. I put in this hole a few cells, 100 cells, and after 3 days if you put the tomato, this part, on the Petri plate, you can see Salmonella here. You have billions of cells. And as you can see here, the tomato is perfectly fine. It's not rotten, okay? So that's why people get sick with tomato, but also other vegetables. It is quite new because in the last 15 years, scientists tried to identify human pathogen. They started to identify as vegetables as a new environment for human pathogens. So human pathogens can survive in vegetables as well, like in the human body. This is the growth curve of Salmonella in tomato. This is the increase in proliferation. That means if you put one cell at the beginning, after 2 days, you will have 10,000 times more than initially known. So it's a huge amount of cells, okay? You see two curves here, and these are the days, okay? Here is the increase of proliferation, and these two curves are, one is the Salmonella 1428, is the same we use in the lab, which is a little bit less virulent, and this is a cocktail from a real outbreak. They behave very similarly, like in the lab, in terms of Salmonella proliferation in tomato. So we decide to use 1428 for our research because it's less virulent and because it's very easy to manipulate the genome. We can easily manipulate this, okay? Delete genes, add some genes, so we can really modify the genome of Salmonella 1428. During my research, we studied this interaction from several points of view. We studied environmental factors, if agronomical practices can help protection of Salmonella proliferation. And I showed this part during the last seminar for the Stephen Cancer, yes, in the cancer laboratory. Today, I would like to focus on the genetics part, which is a little bit more technical part. So that's how we studied this interaction, Salmonella tomato. We studied large deletion mutant library. Basically, we got the Salmonella and we cut a large part of genome and then we tested on tomato. Okay? We use a GFP fluorescent probe screening. This is a technique that allows you to identify genes when the genes activated with Salmonella into the tomato. So you put Salmonella in tomato, we use this technique and you can follow the expression of the genes. This is important because we are interested to see which genes are activated during the interaction with the tomato. We developed, well, we didn't develop, we use the Rivet system, it's gene-reported technology. So this technology allowed us to follow the expression of the genes during the days. Is in real time, we can just in real time follow how a gene is expressed during the interaction, okay, with tomato. And then we analyze the entire transcriptome profile of Salmonella during the interaction of tomato. So the entire genes of Salmonella during this interaction. From the tomato side, we did the same. We studied all the genes activating the tomato during the interaction with Salmonella to see if we can identify some connection. And then we screened tomato mutants. This is another important part. So we had transgenic tomato with some key genes, and then we tested how this tomato behave with Salmonella. So it's quite a lot of information. And so first, let's start with a very easy information. The effect of the cultivar tomato on Salmonella proliferation. When we are talking about genetics, we talk about genotypes, and different cultivars reflect different genotypes. Okay? So at the beginning we say, let's try 3 different cultivars. When you go to the grocery store, you have many different varieties of tomato. Our first question was, do the varieties react the same? Do they have the same infection with Salmonella? So we tested 3 varieties. This is the increase of Salmonella proliferation. As you can see, the variety Bonnie Best is less conductive than the variety B. So basically, you can find tomato more resistant to Salmonella. Okay? We do not have this variety in UK, but with my students we are testing the UK tomato. Okay? Because we need to repeat the same, and we need a system. We already have identified a more resistant variety here in UK. Okay? After these 3 varieties, we say, well, let's test all the varieties known. So we tested 35 varieties of tomatoes to identify which is the variety more resistant. In this variety, you may have important genes to prevent Salmonella proliferation. So you see, for example, if you get this variety, Glacier, okay, or let's get Glacier, for example, this variety. You have 2 bars. One bars are green tomatoes, and the other bar is red, so unripe and ripe tomatoes. Again, you have the increase of Salmonella proliferation here, and all the varieties. So, what we are looking for, something common. What they can have in common, the more resistant varieties. For example, you see this variety is less susceptible, is more resistant. This is extremely susceptible to Salmonella. This variety is less, this variety is less, this is less, this is less. So, which genes they may have? Can we get a gene marker for the breeders, for example? That's our main question. And we found one characteristic in common with all of these tomatoes. So the tomatoes were less proliferating, this one, and this one. They are small. So this is the first, this is a phenotype that reflects a genotype. So we have some gene behind the sides. Okay? So small tomatoes are more resistant. Here is the statistics. Okay? Here are all the variety. We identify like cherry and not non-cherry. These are infected with Salmonella 1428, Salmonella infected with an outbreak cocktail in green and red. As you can see, the cherry are always less susceptible. So in cherry tomato, Salmonella proliferate less.
But also in the US, they kind of genetically engineer these tomatoes to grow larger?
Not really, yes, but it's still breeding. It's not a transgenic plant. They are doing yes, but in the old fashion. Yeah, it's not transgenic at the moment. No. So then we say, okay, if the small ones are more resistant, and the reason may be because the ripening process is different from the small one to the biggest. The smallest, they ripe from inside to outside. So when you get small tomato, they start to ripe inside, and then they move outside. So maturity seems to be involved, and it is because if you try to infect a green tomato, you see the proliferation of Salmonella is less than a ripe tomato. And then we have a partially ripe, these tomatoes are a little bit green and red. So green is more, green tomato is more resistant. Now okay, let's go here. During tomato maturation, you have, there is an important hormone, which is ethylene. Okay, so ethylene has to be involved in some way because we saw that maturity is important. So the question is, can ethylene be involved in this process? And that's why we tested transgenic tomato with impyrate in ethylene synthesis. So what you have here, this is our control. It's green, but if you treat with ethylene, it gets, you see, red. This is 34 days old. This tomato is 46 days old and 59 days old. Okay, but if you use the mutant, this tomato mutant is homozygote for the gene green, so both the alleles are cut. As you can see, and RIN is involved in ethylene metabolism. As you can see, even if you treat with ethylene, they stay green after 46 days and green after 59. And the same for NR and NR. The only difference is this mutant is involved in an ethylene receptor, and because tomato has 3 ethylene receptors, we just deleted one, so the other 2 were still active. That's why at 46 and 59, these 2 tomatoes are yellow. They are not green, they are yellow. Okay? What we did was to infect this tomato with Salmonella and C. This is the control. Okay, here we have Salmonella proliferation. This is the control, green, red, red. And these are the mutants. The mutant NOR, as you can see, Salmonella cannot proliferate even in very old tomato. Salmonella cannot proliferate also in this ring, as you can see here. And in NOR, we say that is those one involved in the receptor, we have a little bit more proliferation. So definitely there is some effect of ethylene, okay, in Salmonella proliferation, and the mutants are more resistant. Again, if we would be able to select a real tomato for the grocery store with this mutation, we could have an advantage in preventing Salmonella proliferation. So what we did, we say, let's try to provide the ethylene to this mutant during the infection and see if we can restore the original phenotype. This is a ethylene chamber. It's an aquarium, okay? So we call ethylene chamber with a very fancy name, but it's an aquarium very close in which we can put ethylene inside, and we have our tomato. So basically, all these tomatoes are infected with Salmonella, okay? And this is the result after the infection and after providing ethylene. If you just want to follow this panel, which is easier at the moment, this is a isoclase, this is the control, this is the tomato after with the Salmonella, okay, and this is A, so not treated. After, then we treat with ethylene. As you can see, if you treat with ethylene, the tomato gets ripe, it's red, okay? But if you treat with Salmonella, also Salmonella increase. That show, yeah, it's not too much, but it's significant. And you can see, during the maturation, you have an increase of proliferation. Let's go RIN. RIN is interesting because what happened here, we treat with ethylene RIN. RIN doesn't turn red, but Salmonella increase. This first means that the pigmentation is not involved in this process. Okay? If we go in the NOR, you see here, we treat the tomato with ethylene, but interesting, it doesn't increase. And NR, yes, we have an increase even if you don't have red, the development or red color. So what happened between these two is not still clear, but we found that RIN is the vegetable tissue here is a little bit softer. It's more soft than NOR. So probably Salmonella can proliferate here just because the tissue is soft. So what ethylene really does is make the tissue soft. And because now the tissue is softer, green tomato is harder than red one. But because we are treating with ethylene, the tissue, the tomato is not get mature, red, but is getting soft.
That's an increased water content, is it? What's making it softer?
Probably what happened, we think, is the cell wall is basically is not more very thick, but it's getting thinner because we have degradation of the cell wall. So there are, some water can go around, so you have an easier environment for Salmonella to proliferate.
But the music getting thinner as well.
Yeah. So, and just to tell you that the Rin, in particular this, it's already used in industry. Many of the tomato you're eating are heterozygote for Rin, because that increases shelf life. So our suggestion, when we're talking with some growers and other people in Cornell University that are making this mutant, was to select for NOR, which may bring some advantage because the tissue is less soft. Okay. So after this, I would like to talk a little bit about the genetics. So we saw some genetics from the tomato. Okay, so the signalin pathway, ethylene signalin pathway, but also from Salmonella side, we found 3 interesting genes that needs to be, they're very interesting. I don't want just, these are the technique we use to identify these genes. So in one case is a GFP promoter library, as I told you before. Basically, if you are not familiar with this technique, is a technique, basically you have a lot of Salmonella, and when you put Salmonella in the tomato, Salmonella, only the genes that are activated, basically they turn fluorescent. And then every time it turns fluorescent, you can catch it and analyze the gene. Okay? You can use this machine, and at the end we identify several genes activated during the interaction. And today I will focus only on YIHT, and using another technique, basically very large deletion mutants. So these are mutants with a larger part of the genome deleted. We identified 2 important genes, RCSA and ZRCSB. Going to talk about a little bit the genetics of these and these 2, which are very interesting candidates, and we proved that if you deleted this gene, Salmonella cannot proliferate more in tomato. Okay? But it can grow in other environments. And finally, well, I don't know if to focus on this, but this is the rivet reported technology, how we are able to follow expression of a gene in vivo. Basically, this is Salmonella genome. You want to follow, for example, this gene, okay, the expression of this promoter. What we need to do is just put a resolvase, which is an enzyme, after the gene of interest. And then in a neutral part of the genome, we need to put a tetracycline-resistant cassette with 2 RES sequences. We will see that these RES sequences and the resolvase will basically let us to follow the expression of the gene, because this is, for example, is the molecule that expresses the gene. So we have the expression of the gene, but also the expression of the resolvase. So the resolvase goes here, it cuts the tetracycline resistance, and basically you can follow the expression of your gene of interest by following the deletion of tetracycline resistance. Looks like complicated, but extremely convenient when you work in a very complex microbial community, because just on tetracycline base replate, they understand it is extremely useful. Okay. So basically, you calculate the resolution is the percentage of cells that are tetracycline sensitive. Okay, and then we can follow the expression of the gene. The first gene we studied was YHT gene. YHT is here. This is the genome of Salmonella, part of the genome of Salmonella. We deleted this gene. And so this gene is involved in capsule formation. This is the capsule. This is Salmonella. The black part is Salmonella, and this is the capsule. So we found that this capsule is extremely important during the interaction with tomato. This is the expression of YHT using the rivet system. Okay, so basically 0 means is not expressed, 100 means is you have a full expression. And this is we study in many different cultivars, okay? Again, we have mature and immature tomato. As you can see, in green tomato, so the immature tomato, the expression of YHT is very high. So, when Salmonella enter in green tomato, it starts to express YHT at very high level. Basically, it is building capsule. So, because this is important, we remove YHT and we tested the deletion mutant for YHT on tomato. And we are expecting that because here is so high, it has to be very important, so the mutant shouldn't grow in green tomato. And that's what we found. To study proliferation of Salmonella, we used a competitive index, which is, we studied the fitness, how is equation to identify how the bacteria proliferate in tomato. And if the number is extremely negative, that means it cannot grow. In this case, the delta YHT, delta means it's deleted, okay? As you can see, it's very negative, that means Salmonella cannot proliferate in green, but that's obviously because the expression of this gene is extremely important. And this is interesting because in a red tomato, Salmonella is around, you see the proliferation is around 0, that means if you delete it, it doesn't care. But that's confirmed by this data, in which in red tomato, the expression of this gene is very low. Okay? So this gene is extremely important in green. And that may have a sense because, as you know, tomato has an immune system, plant has an immune system, plants have an immune system, and the immune system in green is more active rather than in red tomato. So basically, plant is still fighting against the Salmonella. This is another interesting aspect about the presence of tomato metabolites. We were thinking, can we identify some tomato metabolites able to repress YHT? Because if we can identify, we can develop a tomato rich in some compounds and can repress the expression of YHT, so it can prevent Salmonella proliferation in green tomato. So we tested several compounds. This is the expression using River Reporter. Okay, so 0 or 1 means totally repressed, and 100 is full expressed. In this case, the best value is 31. I would like to show this linoleic acid. This means that linoleic acid is able to repress YHT. We tested each compound we can find in tomato. Okay, so linoleic acid is protective. Basically, it represses capsule formation in tomato. And basically, if you have a variety of tomato full of linoleic acid, it helps you to prevent Salmonella proliferation. The last part of this presentation is talking about RCSA and RCSB, the other two genes we identified in our screening. RCSA and RCSB are kind of important genes in Salmonella. They are part of two-component system which react to osmotic stress or membrane perturbation. Basically, you have, for example, acidity of tomato, and so we have two proteins, like the two-component systems. So we have phosphorylation, okay, and then we finally have the phosphorylation on SRB, which is a dimer with RCSA. So both they are phosphorylated, and when they are phosphorylated, they promote cholinic acid. You see capsule formation, and through the repression of RPRA, basically they activate this extremely important gene, Salmonella metabolism, because this gene regulates biofilm induction. So what happened here, when you put Salmonella in tomato, they start to activate capsule formation, biofilm induction, and they repress flagella synthesis, obviously. Okay. So we found that if you delete these genes, Salmonella cannot proliferate more in tomato because it cannot build capsule and biofilm. This is the expression using the resolution of RCSA, RCSB. As you can see, the expression is extremely high. So after just 1 day after the infection, it's fully expressed, 100%. So Salmonella, when Salmonella enters in tomato, it's just start to express RCSA, RCSB at the highest level as possible because Salmonella want to build biofilm. And this is the analysis of the mutants. So we did the lesion mutants. So what we did here, we had the wild type. This is for unripe tomato. This is the mutant on RCSA. It's a little bit less, the proliferation, but it's significant. This is the complementation. So we put the gene back in a plasmid. As you can see, the complementation goes up. If you delete RCSB, there is no question, Salmonella cannot grow at all. As you can see, is this one. If you complement RCSB, so you put the gene back, you have a partial complementation. It can be because you are always modifying the bacteria, so it's not the original gene. And this is the double mutant, no growth at all. Similar for ripe tomatoes, particularly RCSB, complementation, and double mutant. So these two genes are important, and then I left. I left Florida. So now we are here, and that's where we are at this point. We are finishing this work, it will be soon available, but that's our starting point. This one is extremely important, and also YHT. This is another aspect we want to study here. This is the RNA-seq analysis, which confirm the presence of RCSB, RCSA. So they are highly expressed, so they confirm our preliminary experiment. So I'm just concluding. The ethylene pathway is involved in proliferation. RCSAB and YHT support Salmonella proliferation, and linoleic acid repress YHT, so capsule formation. So this is our future: develop green or nor tomatoes for the generation of tomato more resistant to Salmonella. Develop tomato with a high content of linoleic acid, and then identify tomato genes, so genes marker, able to repress RCSAB and YHT. Of course, this is okay, so I would like to thank you. These are the grants, and these are Dr. Teplitsky. This is absolutely genius. He is my age. Okay. He's Ukrainian. Yeah, but he's I don't know. He'll be. Well, then we have Dr. This is the agronomist and undergrads, and these people from Cornell University are the people making the transgenic tomatoes. Okay, done.
Are there any questions for Max? I know I would like to ask what were the outcomes of those 900 people who were infected in the States? Did anyone die?
I don't think so, but no, general Salmonella is not lethal. It's, and also because we have antibiotics most of the time. Yeah.
Okay, so could I, sorry, I'll ask the public health question. You'll ask really a difficult question.
No, it's not difficult. When you said you test your tomato cultivar, so all these tomato cultivar are recent cultivar. Have you tried to test with any more, I would say, wide tomato, I won't say cultivar, I don't want to say cultivar, but wide type of tomato to see how Salmonella interact with something that is less domesticated?
Yeah. Hard question. So let me go back here. All these tomatoes are really wild. The reason is because the grant was funded by, part was public, but part was funded by industry who were interested in identifying, cultivar you can sell. So that's actually interesting. We should ask people, probably from Cornell, they may know some original cultivar, yes, because we don't have here, no. Just to tell you, for example, Florida 47 is the last new brand tomato you have in Florida. It's like this, and it doesn't taste, that's not the main problem, but it's beautiful, but it's just, and we don't have any wild.
Because more white tomatoes, there is more type of white tomatoes, they are not red at all. And
Yeah, right. But here we have no red tomato. Sunny's gold is because it's yellow.
yes, it's yellow because it already has been so selected that we...
I don't know, the first tomato they brought in Europe were yellow. But I don't know, this is gold, but we have many colors here. For example, Body Blucher is extremely red, but we have brown tomato. One of these one is, Kumato is brown, just brown. Because it's dependent on the association chlorophyll and anthocyane pigmentation, so you can see different colors. Colors is not involved because we have extremely high proliferation in all the color range.
No, it's not about the color, it's more about
The
the
answer.
content of what sugar, type of sugar within and structure of the
Yeah.
tissue inside the...
Yeah, definitely. It can be. Yeah. No, it definitely can be. Actually, it would be very interesting to analyze some ancestral tomato. Oh, yes. They may also have some very strong response defense. Yeah.
Yes, well, yes.
Actually, they may have, probably they have stronger, actually. Yeah.
So, you can correct me if I'm wrong, but I don't recall that we had in Europe outbreaks of salmonella from tomatoes. I'm not saying we haven't, maybe we haven't detected them, but I mean, this was a huge outbreak, this particular one.
Yeah.
Was it just luck that it was so large and therefore detected? Are we missing outbreaks from vegetable? Because, I mean, the mechanism of infection and virulence is pretty, generic, I think, what you've shown. So
It is.
are we missing a lot in Europe?
I don't know. I can say that it is generic.
Yeah.
Because people working with sprout, people work with lettuce, people work with melon, cantaloupe, cucumber. We are specialized, every person has his own vegetable. But yes, they are experiencing very similar gene. And dysenterics, they find plants are really a good environment for them. So they are sharing the same genes. So yes, so in tomato is not really common, but in lettuce and cucumber is extremely common for several reasons. Cucumber are eaten raw, for example. Tomato may be cooked. And also lettuce is a big problem. And so, yeah. I don't know in Europe. I don't think so. I don't think we are missing nothing. Or we have stronger regulation, but we have outbreak of vegetables in Europe.
Yes, no, we do.
We have, yes, we have. Vegetables, yes. Tomato, no, at the moment. But vegetables, yes.
Could you just, sorry about that, thinking about why we don't.
Yeah.
Is there any way to detect how salmonella has been introduced to the tomato plants in the first place?
That's this part, the proliferation. That's we are talking about manure. So according with regulation, UK, Europe, UK, and the US, you cannot put manure on the field. You need to compost it before, because the composting process decreases the salmonella proliferation. It doesn't mean that kill completely, because you are not sterilizing something. But so if you have a good compost, you have an extremely low amount of salmonella. Then something can still proliferate. And also, we should consider that wildlife, birds, animals, they can enter in the field, and you never know. what they can do.
But so do you think in that case that the move towards sort of the fashion of more organic vegetables has led to
Definitely.
salmonella
Definitely,
being more
yes.
likely to be?
Yeah, if you use manure not properly composted, yes. No, that's absolutely true. I mean, manure is full of salmonella or other bacteria without considering resistance to antibiotics. That's another field. But
So are the regulations for use of manure in organic different from non-organic?
Well, in organic they don't use the chemicals. So.
No, I know that, but they don't compost the manure?
I think the regulations for industry,
Uh-huh.
when they use a large amount of manure, have to be composted. I don't know if in private organic garden, but definitely using manure in the organic garden, yes, it's more dangerous. Obviously, it's full of enterobacteria manure. Yes, that's. Yeah.
But you can isolate the salmonella from the soil. From
Yes.
these places.
Yes, salmonella can survive in soil for 200 days.
Okay.
So it's very long, yes. That's another interesting aspect, because also we were thinking about salmonella. If you put salmonella outside the human body, it just dies. No. If salmonella can find the proper environment, can survive properly, it's a gram-negative cell, it's very delicate in some way. Salmonella, like E. coli, are very delicate cells, but they are microscopic. So sometimes a small grain of soil has enough to protect them for many, many days. And yeah, there are evidence up to 200 days. After 200 days in soil, no one had never found enterics.
Don't you find the salmonella elsewhere in the plant?
Yes.
You can isolate it from any other cells, the
Yeah.
leaves from the stem.
Yeah, stem, salmonella.
Okay.
It's not very clear, but yes, this is salmonella. Yeah, salmonella can go through the stem, can move through the plant, according to this experiment. We didn't do this experiment. We did this one. This is the
So for 200 days in the soil, in the plant, it can proliferate and increase that way.
Yeah.
So can you actually establish, can you colonise soil simply through reproducing in tomatoes? Because the tomatoes will die and then that could recolonise the soil.
Yeah.
So it could be a self-establishing.
That's interesting cycle, kind of unusual, because we didn't think salmonella about that before. Yes, potentially yes, because salmonella has been identified in the stem. Salmonella can proliferate in the fluid, so if the fluid then, after the cycle, it can re-inoculate the soil. But that's, yeah, it's just the base of ecology. Any cycle of life basically is built on this. So yes, that it can be.
My first suspicion with what you're saying today, I was thinking of the old days of this country when there used to be an association with people who picked fruits, and there was sort of, there used to be rumours, I don't know how much of it was true, not of it, about the pickers urinating on the fruit to create the weight and all sorts of stuff like this. But this is huge amounts, this is mechanised, there's no input like this.
It is, and also, if you have an outbreak affecting people from different regions, north, south, and it's very unlikely that one person is so dirty to infect all these tomatoes. I mean, it can be, definitely it can be, yes. So, but the idea of these outbreaks, that's what we do not understand. It's something that occurs in the field, or it's something that occurs after. It's not clear. That's why this is part that was very important. I didn't talk about this, but this is 50% of our work, in which we studied agronomical practices to prevent salmonella proliferation in the field. So we tried different level of irrigation, different level of fertilization, different level of treatment with chemicals in the field. So we analyzed thousands of tomatoes, and at the end we found that if you use less nitrogen, it's more protective. If you use less water, it's more protective. And in particular, if you harvest during extremely hot and dry days, and if you harvest in very humid days, it's more protective. So humidity and rain prevents salmonella proliferation. Looks strange because you think, well, if there is a lot of water, I would expect it grows more. No, because in the field, when you have a lot of water, you are also selecting the microbial community living on tomato. And because these are it's a phytobioma living on the tomato, they are more selected to survive in vegetables, and they will outcompete salmonella.
I mean, is that part of the issue as well, that, as they say with humans, they're so clean now, that the unexpected exposure to pathogens, actually, there's no competition with.
That's true.
That's part of it too, is it?
Yeah, absolutely.
So can I go back to what I asked you? So, which I wasn't sure your answer was a bit too technical for me. So in the US, they're doing various kinds of modifications, right? Genetic, non-genetic, to make fruits bigger, make vegetables, fruits bigger, or to make them ripen at a certain time.
Yeah.
So do these things somehow affect what's happening with salmonella or no?
Yeah, they can.
Because your outbreak, the major outbreak was in the US and not really in Europe.
Yes.
So do these processes somehow?
Definitely they can. Because, as also Anne said before, as more you modify your plants to get bigger fruits, and probably these plants, after a lot of breedings, they get stressed. They are not very good. And also consider that in plant, more flowers and fruits you are producing, and more the plant is stressed. Because production of fluid is a defense response against dying. They are producing seeds for new generation. But if the plant is perfectly well, it tries to produce less fluids and use all the energy for the vegetation. This is a typical agronomical practice, because I was an agronomist, I studied agronomy, and when they teach how to cut the trees, they always try to teach you, because to cut the tree in a way to stress the plant. You want to stress it, because if you stress the plant, you produce flower and fruits. Okay, so yes, definitely, this plant, they are extremely stressed, their immune system can be compromised, and that's why we have salmonella proliferation. It would be interesting to check some ancestral tomato. Probably there is a gene bank of tomatoes, and probably they have some ancestral seed. It's good to try. We didn't try. Yes, we can.
But probably there's also something about the palatability, producing more sugars and things, which would give them a lovely culture to grow in.
Definitely. Yeah, because probably the ancestral tomato are not very good.
Yes.
That's why also, yeah, definitely. That's something we can try. Yeah.
Any more questions for Max, or we can certainly take ourselves to the tea and coffee. We must eat the pastries, or otherwise we'll never get them again. Meanwhile, thanks a lot.
Thanks a lot.